Acyl-Resin Assisted Capture, or Acyl-RAC, is a sensitive and reliable method that can be used to detect protein S-Acylation in a variety of biological samples.
This protocol bypasses some of the limitations of metabolic labeling and radiolabeling and allows the simultaneous detection of S-Acylation of multiple proteins not only in live cells but also primary tissues and frozen samples. S-Acylation can regulate a variety of cellular processes such as protein trafficking, plasma membrane targeting, signal transduction, ion transport, and protein-protein interactions. It is important to use freshly prepared hydroxylamine solution with carefully adjusted pH for each experiment to ensure an efficient and specific cleavage of the thioester bond. Visual demonstration of this method is critical for steps like chloroform methanol precipitation. The pancake-shaped pellet obtained during this step should be handled very carefully. It can be fragile, and partial loss of the sample during this step can lead to uneven sample recovery.
Demonstrating the procedure will be Savannah West, a graduate student from our laboratory.
To obtain cell lysates, collect the cells of interest into a conical centrifuge tube and remove any cell debris by centrifugation. Wash the pellet in 5 milliliters of PBS and immediately resuspend the cells in 600 microliters of freshly prepared lysis buffer. Agitate the sample at 1500 revolutions per minute in a thermal shaker for 30 minutes at 4 degrees Celsius.